Hi, it's Elle here, and today I want to discuss some interesting advancements in genetic testing that I have discovered. I have had migraines ever since I was an adolescent, like from 15 to age of 30, and it hasn't gotten easier to deal with it, particularly the hemiplegic migraines with paralysis and aura. And I've had MRIs done ever since I was 32 years old, and they discovered white matter lesions in the centrum ceveola, in the white matter deep portion of my brain, and potentially another lesion in the pons area, which is the brainstem. And so basically, because it's hemiplegic migraine and I have them more often than I should, there could be suspicion of CADASIL, which is small blood vessel disease in the brain. It could lead to TIAs, which is mini strokes, or you could have a stroke, or you could have a heart attack even. And for me, it started off with migraines and then progressed into the white matter lesions. My migraines got worse during my mid-20s and my 30s. And according to the age range for when symptoms of CADASIL start for migraines, it's like mid-20s, 30s, and white matter lesions develop by the time you're 35. You can also get dementia, mood disturbances, stuff like that with CADASIL. I live in British Columbia, Canada, so I originally got the limited genetic testing for CADASIL, and they did sequence with Sanger for exons 1, 2, 3, 4, 5, 6, and 11. So that's called limited genetic testing. And that came back negative. So I went back to my neurologist because I still have symptoms of the migraines and I still have the white matter lesions. Those have not scarred over and gone away like they're supposed to. So these might be permanent lesions in my brain. And with CADASIL, the older you get, the more lesions you get. That's the medical statistics. So I went back to my neurologist and I gave him a letter asking him why I would like to get the full genetic testing, NOTCH3 CADASIL testing, which covers exons 1 to 23. And that's not done in British Columbia. That has to be done outside the province. Application funding with MSP. And I got the funding approval. So I went and did the blood work. 4 ml was collected. 2 EDTA tubes of blood. And basically that was shipped off with a funding approval application letter and paperwork to the Molecular Genetics Laboratory at the Health Sciences Center in Ontario, where they did the full broad spectrum of genetic testing for exons 1 to 23. So they do the coding coverage of the sequence for all 23 exons. And they do it with the next generation technology. They don't do it with Sanger, which is very limited in the coverage of the exons. So it's just next generation technology is more advanced in terms of how deep it could probe all the exons. So that's the difference. It codes everything in the exons. So there's a deeper depth coverage of exons and full exons. That's the major difference that I've noticed. I mean, my genetic test result for the limited genetic testing came back negative and I don't think it's negative. I think I do have potentially CADASIL, so I needed the full genetic test to be done, and that had to be done outside the province. So I just wanted to show what the Molecular Genetics Laboratory does in Ontario, why I think it's a superior lab testing. And I'm at the NCBI NIH government website for genetic testing registry. And I'm going to go into the labs section to type in Molecular Genetics Laboratory, London Health Sciences Center, and it's going to give me all the particular information for the lab and how to search for this test. So I go to this tab, the Labs tab, and I type Molecular Genetics Laboratory, London Health Sciences Center. And it gives me a list of potential labs. So I keep typing. It's not enough. Laboratories, molecular, okay, it needs to be Molecular Genetics Laboratory. And then here it is, Molecular Genetics Laboratory, London Health Sciences Center. So I click on that, and here it gives me the information for it. It says it's Molecular Genetics Laboratory, London Health Sciences Center, Department Molecular Genetics Laboratory, and it gives me the address, 800 Commissioners Road E, room B10, 123. It's at the Victoria Hospital, London Health Sciences Center in London, Ontario, Canada. It gives you the phone number, the fax, the email, online contact information, where to download the requisition form. So very right away when you type this in, it gives you a lot of info. But you need to go down to the conditions and tests area, scroll down to cerebral autosomal dominant arteriopathy with subcortical infarcts and leukoencephalopathy. That's the full name of CADASIL. So you need to scroll down to that, click on it, on the test, test one test, and here you will see it says CADASIL, and it says Molecular Genetics Laboratory, London Health Sciences Center, Canada, and it tells you that it does the deletion duplication analysis, which basically measures the amino acids in each exon, whether there's deletion or duplication of the amino acids in the exons 1 to 23. So that's what this is. And the sequence analysis of the entire coding region, this means that they do full coverage coding of exons 1 to 23. And they can also do targeted variant analysis. So if you have some kind of a mutation that's less known and they need to look deeper into it, that's what this is for, as far as I can tell. And the reason why I chose the Molecular Genetics Laboratory in Ontario is because their next generation technology has a high depth coding precision in that they look very deep in your exons for the scanning of the whole exons from 1 to 23. So there's 90 to 100% success rate in terms of percentage, how accurate it is. So that's why I chose this laboratory over Athena Diagnostics, because it doesn't do the deletion duplication analysis. Athena Diagnostics in the States only does the sequence analysis of the entire coding region, and Athena Diagnostics is pretty good. But since I'm in Canada and I also want the deletion duplication analysis done, I selected the Canadian Molecular Genetics Laboratory, and it's the only one in Ontario that does the full NOTCH3 CADASIL testing. The other provinces have limited genetic testing of exons 1 to 6 and 11, I believe. So I'm going to go and look deeper into the testing of this MGL in Ontario. So I click on CADASIL, and it tells me the overview of what it covers in terms of methodology for testing. It says deletion duplication analysis, next generation NGS technology. That's what this is, what I was talking about. It's equal to the massively parallel sequencing, MPS, and it's done with the Ileana MESEC, NextSec. That's what the technology is for the testing. That's for the deletion duplication analysis, and then the sequence analysis of the entire coding region of exons 1 to 23. That's what NOTCH3 means. Sequencing of all the 23 exons. And it's done with the next generation NGS technology. Again, equal to massively parallel sequencing, MPS. And it's done with the Ileana MESEC, NextSec, and also the targeted variant analysis. It's done by directional Sanger sequence analysis. And this can be very advanced also if it's testing for, the deeper mutations that could be had. But if they are using Sanger, then they do need to sequence all 23 exons. And that's done with the Applied Biosystems 3730 capillary sequencing instrument. And again, this is the gene that is being sequenced in NOTCH3. Placement on the gene is 19p13.12. That's the specific exons that they're targeting, where exons 1 to 23 is in NOTCH3 when they're doing testing for CADASIL. And then if you go to how to order, it describes the testing strategy itself, which this is what I found impressive. And I'm going to read it and explain why this is impressive to me and why I think it's advanced genetic testing, why I chose the Molecular Genetics Lab in Ontario. So it says all coding exons 1 to 23 and 20 BP of flanking intronic sequence are enriched using an LHSC custom targeted hybridization protocol, Roche Nimbogen, followed by high throughput sequencing Ileana. Sequence variants and copy number changes are assessed and interpreted using clinically validated algorithms and commercial software, and they list what they use: soft genetics, NextGen, NextGene, geneticist assistant, mutation surveyor, and Alamut Visual. All exons have greater than 1000x mean read depth coverage, so they have a high depth coverage. This is very good if you see this. With a minimum 200x coverage at a single nucleotide resolution. Again, this is very good. 200x coverage is very good of the nucleotide resolution within the exons. This assay meets the sensitivity and specificity of combined Sanger sequencing and MLPA copy number analysis. All variants interpreted as either ACMG category 1, 2, or 3, pathogenic, likely pathogenic, VUS, are confirmed using Sanger sequencing, MLPA, or other assays. So they will use other forms of sequencing if they think it's needed, if they need to go very deep. So they do the NGS, plus they'll use Sanger sequencing and MLPA if needed to do the full coverage of exons 1 to 23. And I like that. So they use a broad spectrum of tools for whatever they need for the CADASIL testing, depending on what kind of mutations may appear in the exons. And there's wide ranges of mutations or variants of deletion duplication. So there's a few degrees of things that could appear in terms of how to diagnose CADASIL. It could be a major mutation, it could be a submutation, or it could be a variant of deletion duplication of the amino acids in the exons. And they say ACMG category 4 and 5 variants, likely benign, are not reported but are available upon request. And test services include clinical testing, confirmation of mutations identified previously, confirmation of research findings, custom prenatal testing, and custom mutation-specific carrier testing. So that's very important. And the test order code is CADASIL NOTCH3. That's what's on the requisition form. And then you go to indications, and it tells you what the CADASIL clinical summary is. CADASIL cerebral autosomal dominant arteriopathy with subcortical infarcts and leukoencephalopathy is characterized by mid-adult onset of recurrent ischemic stroke, which is, or TIA's mini stroke, cognitive decline progressing to dementia, a history of migraine with aura, mood disturbance, apathy, and diffuse white matter lesions, and subcortical infarcts on neuroimaging, which could be an MRI, a CT scan. Those are the neuroimaging tools that are used to look for lesions in the brain. And it also describes, I found clinical validity interesting. The results of the clinical validity studies demonstrate that a NOTCH3 mutation is found in a high percentage of patients with a clinical diagnosis of CADASIL, with studies reporting a clinical sensitivity of 90% to 100%. And that's what the NGS next generation sequencing tool does. It's 90 to 100% accuracy. And they continue to say limited data on specificities from testing small numbers of healthy controls, and no false positive NOTCH3 mutations have been reported in these populations. The diagnostic yield studies report a variable diagnostic yield ranging from 10 to 54%. These lower numbers likely reflect testing in heterogeneous populations that include patients with other disorders. So that's interesting. They give percentages of accuracy for testing with the NGS next generation sequencing, which is 90 to 100% accuracy. That's another reason why I chose the molecular genetics laboratory in Ontario, because next generation sequencing technology provides better accuracy. It does provide better accuracy rates for diagnosis of CADASIL than Sanger, which is 80 to 85% accuracy. And then it goes to the methodology tab, and it explains again the what I already discussed before, that they do the deletion duplication analysis, sequence analysis of the entire coding region, targeted variant analysis. So you can scroll through the tabs and take a look for yourself what the Molecular Genetics Laboratory in Ontario does. It gives you performance characteristics. And it says availability, entire test performed in-house, so it's done in their own laboratory. It's not sent out to third party. Analytical validity, this assay meets the sensitivity and specificity of combined Sankar sequences and MLPA copy number analysis, 99%. So next generation sequencing is just as good as Sanger and MLPA analysis. And it says proficiency testing. Is proficiency testing performed for this test? Yes. Method used for proficiency testing, interlaboratory. So that's interesting too. They do everything in-house in their own laboratory, and they meet all the standards that they need to do this testing. And if you look at the interpretation tab, it provides the same information as I was discussing. It's greater than 1000x mean read depth coverage with a minimum 200x coverage at a single nucleotide resolution. So that's pretty good. And it says what software is used to interpret novel variations. Soft Genetics, NextGene, Geneticist Assistant, Mutation Surveyor, and Alamut Visual. So I found this important because when I went to Athena Diagnostics website through genetic testing registry, in terms of their overall description of their testing, I didn't see all of this description information. I tried looking for it, but I just didn't see what kind of tools they use, and I found I need all the information provided here. So that's why I chose the Molecular Genetics Laboratory for Ontario. I just need all the info that I could find. So it says, are family members with defined clinical status recruited to assess significance of VUS without charge? No, they don't provide that info. Will the lab recontact the ordering physician if variant interpretation changes not provided? So I guess if you need more information on that, you need to call the lab. So that's the interpretation tab, and laboratory contact gives you robust contact information. It says what the laboratory information is: Molecular Genetics Laboratory, London Health Sciences Centre. Laboratory director is Peter Ainsworth. Lab director Joan Knoll. Lab director Tony Rupar. Lab director Bakim Sadikovic, also lab director. So they have a few lab directors. Laboratory contact for this test, Alan Stewart. Informatics staff phone number, email, and they give the laboratory contact, the address of it, and the various laboratory services offered, which I discussed already. So that is the information about the genetics laboratory, Molecular Genetics Laboratory in Ontario, that I selected for my gene testing for NOTCH3 CADASIL, because I was able to find a lot of information on NIH about it, and it has pretty advanced technology genetically, so their depth coverage is very good for sequencing exons 1 to 23, which is, for me, that's pretty important. Yeah. So if I go back, because I actually want to search the lab section again for Athena Diagnostics and just show you the difference. And Athena Diagnostics is in the United States. See, I typed in Athena and it gave me the link to it for the description. And again, same thing here. It tells you its address in the States, Athena Diagnostics, Inc., 200 Forest Street, 2nd floor, Marlborough, Massachusetts, United States, and phone number. And again, you have to go into the conditions and tests and scroll forward. Let's see, they have a lot of interesting testing again that they do. Cerebral autosomal dominant arteriopathy with subcortical infarcts and leukoencephalopathy. And I studied Athena Diagnostics as well, and I found that they have a lot of gene panels for epilepsy, for migraines. So they have quite a robust set of genetic panels, which I found it to be impressive just because they didn't have the duplication deletion for CADASIL, and they're in the United States since Canada has the Molecular Genetics Laboratory in Ontario. I selected Canada to do it in Canada, but if you're in the States, you would choose Athena Diagnostics. So you would go to click on, and it says 4 tests. Yeah, so you click on that, and they do the NOTCH3 CADASIL sequencing test, and it tells you Athena Diagnostics, United States, and it says that they do, for this specific test, they do the sequence analysis of the entire coding region. That's next generation sequencing, that's what it's done with. But they don't do the deletion duplication for some reason for NOTCH3 CADASIL. They also have epilepsy advanced sequencing and SNV evaluation, epilepsy and migraine. And in that one, they do deletion duplication analysis and sequence analysis of the entire coding region. I don't have epilepsy, so I only need this done, and I was able to find laboratory to do it in Canada. They also do epilepsy advanced sequencing and SNV evaluation, and again, that does deletion duplication analysis, sequence analysis of the entire coding region, and they also have complete CADASIL evaluation. So it's not just CADASIL, there's CARASIL and CASASIL as well. And they do sequence analysis in the entire coding region for that. So I'm going to click on the NOTCH3 CADASIL sequence testing just to show you what that looks like. And again, this is the overview. So the genes that it's testing, NOTCH3, placement 19p13.12, where exons 1 to 23 are. And it says methodology, sequence analysis of the entire coding region, next generation NGS, compares to massively parallel sequencing, MPS. So I don't see deletion duplication for NOTCH3 CADASIL for this. I only see the sequence analysis of the entire coding region. For me, deletion duplication and sequence analysis of the entire coding region was important. That's why I chose the Canadian Molecular Genetics Laboratory. I could have chose, out of province also means out of country. I could have chosen Athena Diagnostics, but then I would have had to do conversion from Canadian to US money, and it would have been more expensive. So there were a few factors for why I chose the Canadian Genetics Molecular Laboratory. But Athena Diagnostics is very good. I actually did go directly to their website, and I looked up other gene panels that they do out of curiosity, and they have a lot of different choices, highly recommended. So they're very good too. And if you're in the States, you would go with Athena Diagnostics. How to order information, again, does not provide anything, which, I guess they do provide the test code, which is 1175. Indications, same information as what is on the Canadian Molecular Genetics Laboratory. Methodology, they only say that it's sequence analysis of the entire coding region, so they don't really give you information on what they use to sequence it with, what software they use. I would prefer to know what they're actually using and what the mean depth is. Like, for example, the Molecular Genetics Laboratory in Ontario, it's greater than 1000x for mean depth coverage reading of exons 1 to 23. Again, analytical sensitivity, specificity, and accuracy are 99%, which is very accurate because they're using next generation sequencing, of course. Same for the Canadian Molecular Diagnostics Laboratory. They perform, the Athena Diagnostics performs their NOTCH3 CADASIL testing, entire test performed in-house. Interpretation, they don't provide info on that. And they do provide laboratory information for contacts. Athena Diagnostics Inc., laboratory directors Sat Batish, Canada Data, medical director, that one is lab director, Isabella Carbasi, lab director, Thomas Mullen, lab director, Zanayan Wang, lab director, and their address is 200 Forest Street, 2nd floor, Marlborough, Massachusetts, United States. Laboratory services offered, carrier testing, clinical testing, confirmations of mutations identified previously. So that's what they give for their contact information. And this is how you would look up a lab or labs for any type of different genetic testing. So I thought I'd go over that information because for me, I did have to study all of this. I had to learn what CADASIL is and what symptoms it has and which particular symptoms apply to me and why, and the appropriate laboratory to select, because the laboratories that do genetic testing, NOTCH3 genetic testing for CADASIL, are limited. In Canada, if you want the full NOTCH3 CADASIL testing done for exons 1 to 23, it is the Molecular Genetics Laboratory in the London Health Sciences Centre in Ontario. And in the United States, it's Athena Diagnostics, Inc. in Massachusetts. So those are the two best genetics laboratories for molecular genetics that will do the testing. And it's basically whole blood. They take a blood sample, whole blood, and whichever hospital you do it at or lab, they'll collect the blood work and send it off to the referring laboratory at the molecular lab to do the sequencing of the exons and interpret the results and send it back to the referring physician, which usually it could be your neurologist, it could be family doctor or another specialist that knows something about CADASIL. So this is how the selection process happens to find a molecular genetics laboratory. So I just wanted to share that information and why I found it fascinating, because it took me about half a year of research about CADASIL and the genetic testing involved, and the referral forms, the funding application, because there's several processes in Canada for it. In BC, it's first it's the limited genetic testing. Then if that comes back negative, you apply for MSP funding approval, and then everything is submitted to the genetic molecular laboratory that's selected to do the testing. So that's the Canadian process, and in the United States, it's Athena Diagnostics. Again, I'm not familiar with the United States application process. It's mostly covered by insurance, I think, different insurances. In Canada, it's MSP, not private insurance, or you can pay for it yourself if you don't have MSP coverage, or even, private insurance or whatever, you pay for it yourself somehow. So that's the genetic testing for NOTCH3 CADASIL, full genetic testing sequence. Thank you so much.